Potato wild relatives have a long history of use in breeding. They have been used for several traits such as resistant against nematodes, viruses, abiotic stress, and biotic stresses. Examples of these are Solanum L that displays tolerance against nematodes and frost, and Solanum candoleanum that displays tolerance against nematodes and bacteria. Potato wild relatives have not undergone any domestication bottleneck. This means that they are more diverse in genetic terms than cultivated potato. These wild relatives will continue to be used to adapt potato to new challenges, such as those imposed by climate change and unknown pest and disease outbreaks. Therefore, for being able to use this material, we need to conserve these wild relatives and their habitats. In 2015, together with colleagues from the International Center for Tropical Agriculture, the International Center of Potato, the University of Birmingham, and the University of Wisconsin, we applied a gap analysis methodology to understand the distributions of crop wild relatives of potato, to analyze which places need to be visited for further collections, and also the results of our research enabled us to identify areas where in-situ conservation could be conducted in the future. For doing this, we first identified which are the closest crop wild relatives of potato. That means the species in the primary gene pool, the secondary gene pool, and those species in the tertiary gene pool that have been already used or have a confirmed potential use in breeding. We followed the Spooner's taxonomic revision of Solanum section Petota, and our area of study encompassed the native distribution of potato wild relatives. That means from the United States of America to Argentina. Having this as a reference, we compiled information about existing ex-situ collections of potato wild relatives from online outlets such as Eurisco, Green, and SIP's Biomart portal. In terms of species presence records, we obtained information from online databases like GBIF and PBI Solanum and Herbaria. Then, we used Maxent to estimate the potential distribution of species. We repeated this process for each crop wild relative species to produce individual maps with the likely distribution of the species. We then overlap each of these distributions maps to obtain a single richness map. After this, we were able to apply the gap analysis methodology using the species occurrence records and the distribution maps produced with Maxent. The approach uses three metrics to estimate how much diversity still needs to be conserved in gene banks. Once we obtained these three metrics, we averaged them and divided in four categories to designate the urgency of collecting: high, medium, and low priorities for collecting, and no further collecting for germplasm is required. We also produced maps identifying where further collecting missions could be conducted by estimating a buffer area of 15 kilometers surrounding georeferenced germplasm samples and removing these areas from the species distribution maps. Our study encompassed 73 potato wild relative species, and we produced potential distribution maps for 65. Areas of high species richness were found in Peru, followed by Mexico and Argentina. With regards to the ex-situ gap analysis, we found that 32 species are of high priority for conservation, followed by 20 species of mid-priority, 18 of lower priority, and only 3 are well represented in gene banks. Special cases are Solanum ayacuchense, Solanum olmosense, Solanum neobabilovi, and Solanum salaceanum, which are urgent priorities for collecting since there are no germplasm accessions available. Other 19 species flagged as high priority for conservation have less than 10 active germplasm accessions and consequently very limited representativeness. Areas where further collecting missions for ex-situ conservation could be conducted were identified in Peru, where 20 high priority species are found, Mexico with 4 of these high priority species, Bolivia with 3, Colombia with 2, Ecuador with 2, Argentina, Chile, and Guatemala, each with 1 species. The greatest density of species that need to be collected in Peru is projected to occur in Cajamarca, La Libertad, Ancash, and Huánuco. The adapting agriculture to climate change, collecting, protecting, and preparing crop wild relatives use the results of this study to prioritize areas for collections. The project is now supporting partners in Peru, Brazil, Guatemala, and Ecuador to collect some of the high priority species identified in the study. Sites for active monitoring may be established at areas where several high and medium priority species overlap. Habitats within protected areas or undergoing significant disturbances can also be considered adequate to promote in-situ activities, such as the habitat of Solanum rhomboideae, a limited habitat increasingly exposed to infrastructure expansion and overgrazing. Sites for in-situ management not only offer insights about the evolution, dynamics, and ecology of potato wild relatives, but also the responses of these plants to threats and climate change. Collecting samples of the priorities here identified, monitoring and managing wild populations in situ together with an expanded knowledge of these wild relative species are key steps to ensure the long-term availability of the genetic diversity of a critical crop like potato. This work was undertaken under the project Adapting Agriculture to Climate Change, Collecting, Protecting and Preparing Crop Wild Relatives, funded by the Government of Norway. The project is managed by the Global Crop Diversity Trust with the Millennium Seed Bank of the Royal Botanic Gardens, Kew, and implemented in partnership with national and international gene banks and plant breeding institutes around the world.